Inside a nest beneath the Mediterranean soil, one mother is doing something that should sound biologically impossible. She lays sons that belong to her own species. Then she lays sons that belong to another species, one that split from hers more than 5 million years ago. They do not merely look different. Their nuclear genomes are different. And she needs both. Without the second species, her colony cannot produce its workforce. This is not a metaphor, and it is not a laboratory trick. In 2025, researchers combined field collections, genome-wide data, egg tests, and long-term colony observations to document a reproductive system not previously known in an animal life cycle. They had to give it a new name, xenoparity, the birth of another species. But the strangest part is not that this queen produces a foreign son. It is where his DNA comes from. The queen is an Iberian harvester ant called *Mesa iberica star. Like other ants, her colony looks less like a family and more like a living city. Queens reproduce. Workers collect food, defend the nest and care for the young. Males carry sperm into the next generation. But when researchers examined this species, the family tree refused to make sense. The mystery began with hybrid workers. Across parts of Europe, workers found inside *Mesa iberica's nests carried a mixture of two genomes. Their mothers were *Mesa iberica star. Their fathers belonged to another harvester ant, *Mesa structa star. Hybridization alone is not the impossible part. Different animal species sometimes mate and produce hybrid offspring. The impossible part was geography. Some *Mesa iberica's colonies lived far beyond the known range of *Mesa structa star. The workers still carried the second species DNA, even when no wild population of that species seemed available to supply fathers. So where were the missing males coming from? The team studied genome-wide data from 390 ants representing 5 European species. Every one of the 164 *Mesa iberica's workers in the genetic analysis was identified as a first-generation hybrid. This was not an ancient genetic trace fading through the population. Each worker had one genomic contribution from *Mesa iberica's and one from *Mesa structa star. The colony needed fresh sperm from the other species to make workers. Yet in much of its range, the other species was missing. Then the researchers looked closely at the males inside *Mesa iberica's nests. Some were hairy. Others were nearly hairless. That difference sounds cosmetic. The genome said otherwise. The team compared 62 hairy and 24 hairless male nuclear genomes. Every hairy male grouped with *Mesa iberica star. Every hairless male grouped with *Mesa structa star. Two kinds of son. Two distinct nuclear genomes. One kind of mother. The species were not even closest relatives in the analysis. Their lineages were estimated to have separated more than 5 million years ago. But a genetic result this strange demands more than one line of evidence. The hairless *Mesa structa males carried the same mitochondrial DNA as their *Mesa iberica's nestmates. Mitochondria normally pass through the egg. That mitochondrial signature pointed back to a *Mesa iberica's mother, even though the nucleus of the male cell matched *Mesa structa star. The egg and the nucleus were telling two different family stories. There was still another possibility. Maybe an unnoticed second queen was hiding in the colony. Maybe workers were laying the eggs. Maybe the field samples had mixed together. So the researchers moved into the laboratory. They genotyped 286 eggs and larvae from 5 *Mesa iberica's colonies. 11.5% contained only *Mesa structa nuclear DNA. Then they isolated 16 queens and tested newly laid eggs after 24 hours. 9% again contained only *Mesa structa DNA. Finally, they watched. One colony headed by a single confirmed *Mesa iberica's queen was monitored for 18 months. Among the eggs that reached reproductive adulthood, both hairy *Mesa ibericus males and hairless *Mesa structa males emerged. The mother's genome matched only *Mesa ibericus star. The foreign son's nuclear genomes matched only *Mesa structa star. One mother had produced adult males assigned to 2 species. To understand the mechanism, begin with the unusual genetics of ants. Female ants usually develop from fertilized eggs and carry 2 sets of chromosomes. Males usually develop from unfertilized eggs and carry 1 set, inherited from their mother. These foreign males reverse the expected direction. Their nuclear DNA appears to come from sperm stored inside the queen. The queen's egg supplies the cell, including its cytoplasm and mitochondria, but not her nuclear genome. The resulting male is effectively a clone of the paternal lineage. The process is called androgenesis, development in which the father provides the embryo's nuclear genetic material. The study found sperm from both ant species stored in queens that produced both kinds of male. But the exact cellular event remains unresolved. Scientists do not yet know whether the maternal nucleus is excluded from the egg or removed after fertilization. That uncertainty matters. The evidence shows what genetic result occurs. It does not justify inventing a conscious queen editing DNA inside her eggs. Evolution built the system. The queen does not need to understand it. Now the reproductive puzzle becomes a loop. When the queen produces a male of her own species, his sperm helps produce future *Mesa ibericus queens. When she clones a male of the other species, his sperm helps produce hybrid workers. Queens come from the in-species line. Workers come from the cross-species line. And those workers are essential. They excavate the nest, forage, defend the colony and raise the offspring, including the males that keep the system running. The queen cannot build a normal workforce using only males of her own species. Her colony has become reproductively dependent on *Mesa structa DNA. At first, the dependency may have required mating with wild *Mesa structa males wherever the species overlapped. But dependence on a wild partner limits where a colony can survive. The extraordinary evolutionary solution was to carry the partner's lineage inside the colony. The researchers found 2 versions of the *Mesa structa male lineage. One was wild. The other was a nearly identical clonal lineage found inside *Mesa ibericus nests. Of the 164 hybrid workers whose fathers were traced, 144 had fathers from the clonal lineage. Only 20 had wild lineage fathers, and those cases occurred where the 2 species still shared the same region. The clonal lineage also showed extremely low genetic diversity and a higher genetic load than the wild lineage. Its males were nearly hairless, while wild males were hairy. The authors compare this relationship to domestication. Humans domesticated animals by controlling which individuals reproduced. Here, natural selection produced something far more intimate. One ant species maintains the male genome of another, reproduces that genome through its own eggs and uses those males to generate the workers that sustain the colony. That does not mean the ants made a plan. Domestication is the researcher's evolutionary comparison, not a claim of intention. It means a lineage that once had to be found in the wild is now propagated inside another species' life cycle. Xenoparity means more than unusual motherhood. Inside one colony, different bodies carry dedicated biological roles. A *Mesa ibericus queen produces her own species males. She also produces *Mesa structa males. Cross-species mating produces hybrid female workers. Sex, caste and species are woven into one reproductive machine. The paper calls this a possible 2-species superorganism. That phrase does not erase the genetic boundary between the species. The researchers still identify the clonal males as *Mesa structa star. But the 2 lineages have become so interdependent that neither part explains the colony alone. One species supplies the queen and the egg. Another supplies a cloned male nucleus. Their hybrid daughters perform the labor that keeps both genetic lines moving through time. The colony is not simply a place where two species meet. It is the process that connects them. For centuries, biology has organized life by drawing boundaries: mother and father, individual and colony, one species and another. Mesa Ibericus does not erase those boundaries. It threads a life cycle through all of them. A queen lays an egg. The egg carries her mitochondria. Its nucleus belongs to a male lineage from another species. That son grows up inside her colony, father's hybrid daughters, and those daughters raise the next generation. The discovery is not proof that species mean nothing. It is proof that evolution is under no obligation to keep its categories simple. And beneath the Mediterranean soil, a creature only millimeters long is preserving an entire foreign male lineage inside the body of its own society. If this changed how you think about what an individual animal can be, subscribe to CreatureCosmos. And tell us in the comments, is this one colony, two species, or something evolution has forced us to name for